in order to be able to talk about the microbiome in eczematous skin. I'm highlighting here a series of experiments by a collaborator, Yasmin Belkade's group, to highlight some of the differences that we see in that this not only is the skin microbiome different than other body sites, but that the skin immunity and the skin microbial relationship is also distinct. Here, Shruti, who was a graduate student in Yasmin's lab, she treated SPF mice. These are all experiments in mice with antibiotics. And what we can see in the gut, that after antibiotic treatment, there is a significant shift in the microbial communities, but you don't see those significant changes in the skin microbial communities. Also, Shruti looked at the immune cells that were in the gut, IL-17A, as well as interferon gamma producing cells in the gut, and as compared to the skin, once there was treatment with antibiotics, there was a significant reduction in the interferon gamma and IL-17A producing cells. So there are, in distinct epithelia, there are differential responses to antibiotic treatment, at least in these mice. So Shruti then went on, and in comparing SPF mice and germ-free mice, SPF mice here, germ-free mice, looking at IL-17A producing T cells in the gut and the skin, we see here that there are reduced IL-17A producing T cells in the germ-free gut and skin. However, what she then did was topically mono-associated staph epidermidis in these germ-free mice, and interestingly, there were no changes in the IL-17A producing T cells in the gut. However, in the skin, there was an increase of IL-17A producing T cells in the skin after application of staph epidermidis. So we can see here that skin microbes do influence the immune system, but is there a function, is there a biological result as when you do that? So what then Shruti did was used a Leishmania major infection model. She infected the ears of these SPF mice and germ-free mice, and in comparing these two, you see that there is a local cutaneous response in the SPF mice as compared to much larger than the germ-free mice, but also you see increased interferon gamma producing T cells in the SPF mice as compared to the germ-free mice. So then when she topically associated staph epidermidis in these germ-free mice, you see an increase in the local cutaneous response as well as interferon gamma producing T cells that are in the ear. And these resemble what you see in the SPF mice. But also, here we see in parasite burden that you have a decrease in the parasite burden when you treat or topically associate staph epidermidis. So skin commensals can restore immunity to Leishmania major infection in germ-free mice. So summarizing this part, the host immunity and microbial interactions in skin are distinct. In mice, skin microbes can tune the level of activation and function of skin-resident T cells, promote immunity to pathogens, and drive responses locally that are distinct and independent from the gut flora. But what about human skin immunity and microbial interactions? Unfortunately, we don't have a lot of germ-free humans walking around that we can do these types of experiments, but what we have done is started to look at what are the microbes that are on human skin. So again, this is a background for those who are not as familiar with human skin microbiome surveys. These are highly selected due to limited time that I'm not going to go into all the surveys that have been done. You've seen this figure before, but the reason why I'm showing it again is I want to emphasize that in skin, here in pink, there is a wide variation in the microbiome that you see in skin as compared to the other body sites. And that is because the skin surface is highly heterogeneous. And that is one of the reasons why in this study we selected 20 skin sites. These are distinct microenvironments on the skin surface, but I also selected them because they were sites of predilection for skin disease, specific skin diseases for which microbes have been postulated to potentially have a role. And David Relman showed this figure earlier, so I won't go into it in detail. But relative abundance of predominant bacteria appear to be dependent on the microenvironment. So we've talked about this throughout yesterday and some this morning, how there are carefully defined cohorts that we have looked at. Most of these are adults. We've talked about what happens. There have been the studies by Maria Gloria looking at the neonatal period, but what happens after that. Ruth Lay talked about at least for the gut microbiome, how that can transition in the first 2.5 years. But what about the skin? We don't know that information. In the other next transition, puberty. This is a turbulent period of time for our bodies. What happens as we transition into adulthood? There is something called a Tanner stage. For those of you who are not clinicians, a Tanner stage is used by pediatricians to assess the level of sexual maturation in an individual child. This is based on the development of breast tissue as well as genitalia, pubic hair, but it ranges from 1 to 5, 1 being pre-pubertal, 5 being fully developed adult. So what we have here is we had cohorts of patients that were ranging from 2 to adulthood. So what Julia O did in Julie Segre's lab was take these data and compare them with each other. And based on Tanner stages 1, 2, 3, 4, and 5, we see the striking difference between Tanner stages 1, 2, 3 and Tanner stages 4 and 5. This is from the nares, but we see a similar separation in other skin sites. What drives that? Here we see a relative abundance chart, which you've seen a lot in these talks leading up till now. And here is Tanner stages 1, 2, 3, and then here is 4 and 5. I do acknowledge that the ends are pretty low in these populations, but what we see in Tanner stages 1, 2, 3, that we have increased proteobacteria and more lipophilic bacteria in Tanner stages 4 and 5. That makes sense for everybody who's gone through puberty. You recognize that your body is changing dramatically, and one of the things that changes is the further maturation of our sebaceous glands or oil glands in our skin. So it's very possible this may be one of the reasons why we have more lipophilic bacteria, because they can thrive in that type of environment. So we've talked a lot about 16S taxonomic surveys. We've heard some about the virome and several of the investigators looking at that. But what about the fungal microbiome? So more recently, we've published a project looking at sequencing the fungal organisms on the human skin surface. But in order to do this, we had to go back from the beginning and figure out how do we optimize sample collection. And this refers back to the conversation we had in the open floor session yesterday. What are the protocols to use? Optimizing DNA extraction, we had to deviate from the standard protocols for extracting DNA in order to optimize extraction of fungal DNA using bead beating, opening the fungal cell walls. What primers would we use? There are individuals who use 18S, some who use 28S. There's the ITS region here, ITS1, ITS2, that stands for internal transcribed spacer region. And so based on our analyses, for us, at least for skin, that ITS1 provides more taxonomic resolution. This alludes back to Jacques Ravel's comment yesterday that just because it works for the gut or for the vagina, it may not work for the skin. And so this is for, at least in our experience, ITS1 worked better for taxonomic resolution. So what we did was we had 10 healthy volunteers. I selected 14 sites. These are not identical to the ones that we did in the bacterial survey, because these are, again, sites that reflect sites of predilection for skin diseases for which fungal contribution may be playing a role. So in 10 healthy volunteers, we see here. Then if you look at the relative abundance charts, each of these horizontal lines represent one body site. Each of the vertical bars represents one of the healthy volunteers. And one of the most striking things that you can observe here is that malassezia predominates in 11 out of the 14 sites. That's the purple. That's why there's so much purple. But on the heel, toenail, and toe web space, so on the feet, there is much greater diversity. We have fungi everywhere, but it's just a different population depending on where you are. One of the things that you may notice is that healthy volunteer 7 is a bit different. This individual, again, talking about protocol standardization, our eligibility was no one could have taken an oral antifungal oral antibiotic within 6 months of being sampled for this protocol. And this individual had completed a course of oral antifungals 7 months prior to being sampled. And so it's not clear whether the differences we see are related to residual effects of taking an oral antifungal or whether or not the person's predisposition, they were taking it for a toenail infection, whether a predisposition for a toenail infection represents some difference in their fungal microbial communities. Interestingly, I don't have the data here, but their 16S survey for this particular individual, HV7, was similar to the others. So the bacterial microbiome resembled any other healthy volunteer. But it was frustrating to see so much purple. There's a lot of malassezia, that's not that helpful. But one of the questions we had was, what happens if you speciate? Getting down to the speciation level, how do we do that? Well, we couldn't do that based on the databases that were available. So we had to cultivate our own malassezia and do genomic sequencing to actually populate the databases for us to be able to speciate. So when you do that, you again see there is striking site specificity for skin. Sarkis was talking about how there's specificity in the gut, but also we see that for the skin with regards to the fungal microbiome as well. So we can see in this area here, the external ear canal, behind the ear, the forehead, that malassezia restricta predominates. But in other sites, we have the upper back, the back of the scalp, the inside hip. Those are predominant, but you have Malassezia globosa. So even though they're all Malassezia, there are specific species that we're seeing at these different body sites. So I did mention that we did 16S. So from these same clinical samples, we did ITS1 sequencing as well as 16S sequencing from these samples. And what you see here is interesting anatomically in that those central body sites from the head and neck, or core body sites listed here, they have a relatively limited number of, with regards to richness, relatively limited numbers of different types of bacteria and fungi. Whereas on the arms, here's the palm, the forearm, and the inside elbow, there is a higher richness, a greater richness with regards to bacteria, but still relatively limited for fungi. That's different for the feet. Here, the heel, toe web, and toenail, relatively limited richness for bacteria, but a much greater richness for fungi. So there are these regional differences that cannot be explained just by sebaceous, moist, and dry, and that it's going to require a lot more understanding about human physiology and skin physiology to be able to explain these differences. So just summarizing this beginning portion, the skin bacterial microbiome is highly dependent on the sampled skin site. The neonatal skin bacterial microbiome varies based on mode of delivery. We've talked about that. That there are dramatically major shifts that we see in the Tanner stages 1, 2, 3 versus Tanner stages 4 and 5, and that fungal communities over the skin surface differentially vary from the bacterial microbiome. Now I'm going to shift into what my charge was, into talking about eczematous skin, specifically atopic dermatitis, and then briefly I'll talk about some data from other groups and our group about primary immunodeficiency syndromes and why that's interesting. So for background, those who are not familiar with atopic dermatitis, it's a chronic, itchy, inflammatory skin condition. It is not considered an infectious disease, it's an inflammatory skin condition, yet these individuals respond relatively well to antimicrobials. So there is something going on that suggests a role of microbes. It affects 15% of US children at a high cost, financially as well as socially. The quality of life in these children and in these families is severely adversely affected. And I mentioned before, there is this association with microbes. We observe, and when we take care of these patients, that disease flares are associated with increased colonization and infections with Staph aureus, but also a subset of these patients are at high risk for severe spread of herpes simplex virus infections, and if they come into contact with smallpox vaccinees. There is something that's been termed the atopic march, in that there are 40 to 70% of those with severe atopic dermatitis over time go on to develop asthma and then hay fever. So these three diseases are termed the atopic triad. The incidence of these atopic diseases have doubled in the last 3 decades in industrialized countries, suggesting there may be a possible external factor, and this alludes to some of the comments yesterday by Marty and again today, that it is unlikely that our human genome could change in that period of time and that an external, possibly microbial contribution may be playing a role. Interestingly, in mice, in murine studies, skin exposure to antigens can result in subsequent mucosal sensitization to those antigens, suggesting that if we could somehow modify what happens in atopic dermatitis, could we then go on and abrogate the development or the disease severity of asthma, which has significant morbidity and potential mortality, as well as hay fever. So understanding the triggers of atopic dermatitis may allow us to modify the development of AD and atopic disorders and potentially develop therapeutic targets. So atopic dermatitis is a complex disease. It looks very simple here, but this is not the whole story. But just emphasizing, we've talked about barrier, the gut barrier earlier today, but emphasizing I won't talk about it here, but it is important in atopic dermatitis. The skin barrier, we know if you have a mutation in the skin epidermal protein, filaggrin, it's highly associated with the development of atopic dermatitis, particularly the kind that goes on to develop asthma and hay fever. We know the immune system is deranged in these individuals and that they have extremely high IgE levels and antimicrobial peptides in the skin are reduced. And we've also talked about the microbes, and that is just one component of this complex disease. So our study, we recruited pediatric patients with moderate to severe disease and healthy age-matched controls. I sampled them in characteristically affected sites. As I mentioned before, dermatologic diseases have sites of predilection where we find that, and that helps us in the diagnosis of these diseases. But it typically occurs in the antecubital fossa and the popliteal fossa, that being the inside elbow and behind the knee. We selected the volar forum or the inner forum as a control site because it is less often affected in moderate disease, and it's an adjacent site. We also sampled the nares because that is a site of carriage for Staph aureus. And I sampled them during the baseline flare and post-flare time points, and these are just SCORAD scores, which is a method for assessing severity in these patients. And you can see that over time, their SCORAD increases during the flare and decreases after they've been treated. This figure just demonstrates that when you have more severe disease, that we observe a decrease in the bacterial skin communities. But that drop in diversity is not everywhere. It is very site-specific again. It is at the sites where we see disease appear, the inside elbow and behind the knee, but not at all time points. This in particular are disease flares. These are the true natural history of the disease, where they have not been putting anything on their skin. The blue flares are individuals who put on topical steroids potentially 2 days before seeing us or sometime within the 7 days before they were sampled. So these flares are truly the natural history of the disease. But what are the bacteria that seem to be driving this decrease in diversity? So looking at the genus, down to the genus level, here are the healthy controls. These are the baseline time points for the atopic dermatitis, the flare time points, the no treatment ones versus the intermittently treated ones, and the post-flare time point. And what you can see is that there's a dramatic increase, a significant increase in the pink, the Staphylococci, in the skin of these patients during a flare. But we do see increased staph in proportion or relative increase in some of these individuals. So that was quite a concern. Again, we had to go down to speciation level because there's Staph epidermidis, Staph aureus. Staph epidermidis, again, is a known skin commensal. Staph aureus is a fairly common pathogen. And so it was important to us, at least from the genome level, they're very similar. So we had to know which one was which because it makes a difference, at least clinically. And so what we did, if you focus primarily on the pink, the Staphylococci, and we speciated those, it was reassuring to see that most of the staph we see in the healthy controls, those are Staph epidermidis, Staph hominis, known skin commensals. But what we do observe is, yes, there's an increase in the Staph aureus that we see even at baseline, but it is dramatically increase during the flare. The natural history of the disease, we see increased Staph aureus, but also Staph epidermidis. That was not something we did expect to see, but that is one of the benefits of looking at the whole microbial community with regards to disease progression. And then it decreases once patients have been treated. And one of the questions people often ask, well, you gave them antibiotics, but often these majority of these patients were limited, their treatment was limited to topical steroids, or some of them do take dilute bleach baths, which is like having a small swimming pool in your own tub. So that's what we observed. But what remains a question is this are correlations. What happens to get from this point to this point, or what can we do so that you remain a control, you don't go on to develop atopic dermatitis? Those are questions that remain. We did look at the fungal communities on the skin. Julia O looked at this. These are baseline flare and post-flare for a few of our patients who had atopic dermatitis. And although you see fluctuations in the bacteria, we don't see that type of fluctuation in the fungal communities on the skin. So I'm going to shift gears a little bit and talk about primary immunodeficiencies. There are some cohorts of primary immunodeficiency patients who have eczematous skin disease. And the benefits here are they genotypically have the same mutation. They are monogenic disorders. And some of these patients have atopic dermatitis-like skin eruptions. And these eruptions can be antibiotic responsive again. And so that was one of the reasons why we pursued these cohorts. And asking, do common and rare diseases with similar clinical phenotypes, do they share skin microbiome features? And how does the innate and the adaptive immunity shape the skin microbiome? We haven't been able to ask all of these questions, but these are some of the reasons that we were led down this path. I'm just going to talk about two of these diseases in the rest of the time that I have, talking about hyper IgE syndrome, which have STAT3 mutations, as well as another paper that was recently published by Dirk Jeevers and colleagues looking at STAT1 mutations with chronic mucocutaneous candidal infections. So STAT pathway is important. It's a biochemical pathway, and it's involved in so many things, but these patients are at risk for infections. On hyper IgE syndrome, they have staphylococcal skin and lung infections, candidal infections, and they can develop secondary aspergillus lung infections. So this is a paper I just mentioned by Dirk Jeevers and colleagues, where they looked at STAT3 and STAT1 mutations. It's hard to see, but the leftmost groups, these are the STAT1 mutation patients. The middle grouping are the hyper IgE syndrome patients or STAT3 mutations, and these are their controls. But in general, what you see, just summarizing everything in this little corner, at the genus level, you see increased Corynebacterium species in the STAT1 mutations. You have decreased Corynebacterium species in the STAT3 mutations. You have increased gram-negative